Hello, everyone. My name is Manuela Raimondi. I am a professor of bioengineering at Polytechnic University in Milano, Italy. First of all, I would like to thank the organizers of this conference for inviting me to give this talk, and it's a great honor for me. I would like to introduce myself very briefly. I am the founder of the mechanobiology group at Polytechnic University in Milano, where I direct 2 labs and this wonderful team that you see in the center. I invented some frontier platforms for biological research, MOAB, the Nishoid, and the MicroAtlas that I will tell you about in this talk. I have recently completed a 1-year sabbatical at the Gottardi Lab at the University of Pennsylvania that greatly broadened my research interests and views, and I am very, very grateful to Riccardo for hosting me at his lab. I have potential competing interests and that I have patented all these inventions. Also, together with my students, I founded a startup company that commercializes these platforms. So a primary frontier in cell modeling is maintenance of cell function. For example, we know that mesenchymal stem cells that reside in bone marrow can release factors that slow down inflammation or else differentiate in various cells of connective tissue, such as bone cartilage. So they are extensively studied in hundreds of clinical trials. However, these cells tend to lose pluripotency when expanded in vitro. This problem limits the realism of cell models based on multipotent cells. So how can we recreate in vitro a microenvironment able to maintain cell multipotency during expansion culture, or even induce a spontaneous reprogramming of pluripotency in cells? This would be even better because this is the true definition of a stem cell niche. So we first of all have to make a hypothesis on how the stem cell senses its physical microenvironment, for example, while crawling on a scaffold in culture. My hypothesis was that when the cell modifies its morphology, the nucleus senses such deformation through a direct mechanical link between the cytoskeleton and the nuclear pore complexes. When the cell spreads, the nuclear envelope is strained, and this in turn activates the nuclear pore complexes, increasing the permeability of the nuclear envelope to the nuclear import of signals called transcription factors in the green dots you see in the figure, involved in key master switches that dictate cell function, for example, the transformation of a stem cell into a differentiated cell. So we were able to predict theoretically the diffusive flux of a transcription factor with increasing cell deformation. You can see here the output of a multiphysics computational model of mass transport and cell spreading mapped on 1/4 of a cell. We coupled cell deformation to diffusive flow by assuming a strain-dependent diffusion coefficient at the nuclear membrane. However, to validate my hypothesis, I had to figure out how to experimentally control the morphology of cells, not only on a culture dish, but also in space and time at the cell scale. I needed to isolate the mechanical contribution from the biochemical one, well-defined mechanical environment, stable in space and time. I needed to maintain cells in 3D contact between each other to allow for paracrine signaling. I also needed to monitor nuclear entry of signals that regulate gene expression, so-called transcription factors in space and time. So none of the available scaffolds was adequate for this task. Luckily, around 10 years ago, I met with Giulio and Roberto, 2 physicists who introduced me to 2-photon laser polymerization. This is a microstereolithography technique in which polymerization is induced in hybrid organic inorganic photopolymers based on silicon and zirconium, like this SZ2080 in the figure. A femtosecond pulse laser beam induces 2-photon adsorption in the resin, which polymerizes in a volume very close to the focus. The sample can then be moved in space while keeping the laser on focus, and this allows to literally write the microstructure in the resin with an astonishing resolution down to 100 nanometers. Advantages are that the geometries are totally arbitrary, and this resin is inert, hydrophilic, and so very biocompatible. So with this microfabrication technique, I invented scaffold called the nichoid. So it sounds like organoid, but with an emphasis on reproducing the structural complexity of the stem cell niche. This microscaffold is a microscopic grid able to maintain cell adhesion in 3 dimensions in a very isotropic configuration, maximization of cell-to-cell contact, and allows for physiologic paracrine signaling. We performed a 3D reconstruction of the nuclear membrane pores by scanning transmission electron tomography to estimate the area of the nuclear pore complexes available for nuclear import while cells were cultured inside this microscaffold. So compared to flat, we found that stem cells adhered, these are mesenchymal stem cells in particular, adhered to the internal lattice of the nichoid with reduced expression of structural proteins that form the focal adhesions like beta-actin and vinculin, as shown in this very, very elegant co-localization study on the left by Emanuela of my group. Also, the number, length, and area of the focal adhesions were significantly lower in the nichoid compared to flat cultured cells. So the 3D adhesion configuration induces a significantly different nuclear shape in nichoid culture cells that become very roundish compared to flat culture cells that have a flat nucleus. So we used also fluorescent recovery after photobleaching FRAP to measure to measure the nuclear import of transcription factors, and we concluded that cell spreading was in fact a primary factor determining the permeability of the nucleus, for example, to MyoD, which is a transcription factor involved in mesenchymal stem cell differentiation. We found the transcription factor significantly more localized in the nucleus of cells cultured on flat, so the spread ones. But most importantly, we were able to visualize the fluorescence recovery proportional to the nuclear flow on the entire nuclei in space and time in the 2 conditions, on nichoid and flat. We discovered that in the nichoid, the flow is reduced and also very homogeneous as the particles flow inside from all the directions in space. On flat controls, instead, not only the input flow is faster, but also it occurs from the peripheral areas of the nucleus. So on the left, we can visualize the fluorescence recovery homogeneous on the nucleus. On the right animation, you can see the recovery is very highly dishomogeneous. It mainly occurs on the peripheral areas of the nucleus. So this mechanical configuration able to release the tension from the cell nuclei has a very, very significant effect on cell gene expression. The nichoid promotes spontaneous upregulation of stemness genes in mesenchymal stem cells, specifically the leukemia inhibitory factor LIF, and also cytokines such as interleukin 6 and colony stimulating factor 3, which is highly upregulated, and this promotes function and mobilization of hematopoietic stem cells. So we are seeing this CSF3 gene strongly upregulated also in different nichoid geometries, by the way. This result is coherent with the current knowledge on the architecture of the native hematopoietic stem cell niche, in which mesenchymal stromal cells, the ones in green in the figure, have an arboreal stellate morphology in 3 dimensions with few thin extended extrusions that contact hematopoietic stem cells, regulating primary aspects of their function. So we now have a lot of work ongoing to measure this reprogramming effect of the nichoid in the absence of chemical conditioning on hematopoietic stem cells as well, and also on gene-modified hematopoietic stem cells that are being tested in clinical trials to treat monogenic diseases such as sickle cell anemia, thalassemia, and primary immunodeficiencies. We started working on a cell model that could be translated to the clinic's neural progenitors. We expanded erythropoietin-secreting neural progenitor cells, NPCs, and we found upregulation of the expression of the 3 main genes of pluripotency when cells were expanded in the nichoid substrate. Also, the neural progenitor cells detached from the nichoid, where after being detached from the nichoid, they retain a memory of pluripotency induction with increased expression of neural markers, for example, if re-expanded in suspension culture. Most importantly, the therapeutic effect of nichoid culture cells was significantly improved in an experimental animal model of Parkinson's disease, as shown on the upper panels by the functional recovery indexes that are significantly higher in animals treated with nichoid culture cells. The behavioral data are confirmed by the presence in the transplanted cerebral tissue of a greater number of cells positive to tyrosine hydroxylase, which is a key enzyme of dopamine synthesis. Based on these results, we filed a patent entitled Geometric Induction of Pluripotency. We also looked at the effect of the nichoid on embryonic stem cells that have a potential to repair tissues of any type, so the retina, the spinal cord, cardiac tissue, and all the others. So what we did, we expanded R1 mouse embryonic stem cells in the nichoid without any chemical conditioning. At day 7, cells from smaller embryoid bodies than in suspension culture on flat. This physical containment caused two effects. Pluripotency genes were switched on, and you can see octamer 4 still in the nuclei at day 7, whereas it is totally lost on flat. And simultaneously the downregulation of several differentiation genes, GATA4 for example, but also all the other ones we tested. So thanks to these very good results, we approached large-scale fabrication of the nichoid microscaffolds. We used multi-Fauci parallelization with a spatial laser modulator that basically splits the incoming single laser beam into multiple laser beams, all focused on the sample at the same time. We are able to cover a surface of 1 square centimeter with 10,000 microlattices in only 2 hours of continuous laser writing instead of 20 hours like in 2010. So our ultimate frontier for application of the nichoid is now space. Thanks to a collaboration with Gianni, we are developing a non-frangible version of the nichoid subset for cell culture experiments in microgravity on board the International Space Station. The problem we had to face is that glass, that is the traditional substrate material used for two-photon structuring and also for imaging, thanks to its stiffness and optical transparency, cannot be used on the International Space Station due to the safety requirements. So we performed two-photon polymerization of microscaffolds arrays onto a cyclic olefin copolymer, COC, commercially known as Thermanox. This will open new avenues for the use of the nichoid also on Earth, for example, implantation of cellularized constructs in vivo. Okay, so another very important frontier in cell modeling is to maintain cell function in long-term culture. It's not only to maintain cell function, but also in long-term culture, to recapitulate slowly developing processes in vitro. So we had the idea some few years ago to incorporate in a bioreactor on a microscopy slide, and together with Matteo, I invented the miniaturized optical accessible bioreactor. This is where the acronym MOAB comes from. It's a millifluidic bioreactor with 3 culture chambers built on it, allowing to inspect cellularized constructs non-destructively in fluorescence at various time points of perfused culture. For example, this is how collagen secreted by chondrocyte cells can be visualized on the live cells with an intravital fluorescent laser. We use computational modeling to predict and control convective mass transport of nutrients and oxygen in the system, as you can see here, in the aim to control tissue development in function of these parameters over long-term culture times, up to several weeks. For example, this is a computational prediction of the fluid dynamics environment acting on culture cells subjected to interstitial perfusion in the bioreactor's chambers. So a very slowly developing process is the formation of a bone metastasis by breast cancer cells. So a frontier use of frontier cell culture method is to simulate this very slowly developing process. So endothelial cells and bone marrow stem cells mediate tumor cell proliferation in the perivascular tumor niche, and the MOAB allowed to add this effect of this interstitial flow to the model. We found that interstitial fluid flow upregulates endothelial sprouting. Endothelial cells self-assemble in a loose vascular bed in static culture, and in a more dense vascular bed when perfused, likely by increased convective transport of angiogenic signals. So in this model of the metastatic niche, we measured a significant increase in cancer cell survival without flow compared to what measured with flow, but only when cancer cells were put in co-culture with stromal cells and endothelial cells. Under interstitial flow, cells proliferated less but presented a significantly greater resistance to traditional chemotherapeutic drugs because these agents, like sunitinib, for example, they target proliferative cancer cells. Thus, they fail to inhibit breast cancer cells exposed to interstitial flow, similarly to what we see in animal models. So interstitial perfusion adds realism to cell models used for testing chemotherapy drugs against metastasis, for example. Another very, very slowly developing process is chronic neurodegeneration. We insert neurons with a damage similar to the one induced by Alzheimer's disease, and we culture these neurons for many days under conditioning with neuroprotective factors secreted by bone marrow cells. We could measure a significantly higher viability of the conditioned neurons, suggesting a neuroprotective action exerted by bone marrow cells when maintained in perfused culture for several days. We are now exploiting this possibility to measure paracrine effects of mesenchymal stem cell secretome on pulmonary stromal cells damaged by the SARS coronavirus virus in a similar model. Other mechanisms of neurodegeneration can be modeled by connecting multiple mobs representing separate compartments of a functional body axis, like the microbiota gut-brain axis that my colleague Carmen is modeling in her very ambitious Frontier platform that connects the microbiota, the gut, the immune system, and the brain to recapitulate the effect of microbiome-secreted neurotoxins on the development of Alzheimer's disease. Another slowly developing class of interactions are typical of our adaptive immune system. For example, in a lymph node, dendritic cells in green enter, adhere to reticular cells in yellow and orange, and express receptors of T cells in blue to attract and instruct them. This process of lymph node instruction can be modeled in the MOAB by seeding stromal cells in yellow on a scaffold, then by introducing dendritic cells suspended in the perfused culture medium. Dendritic cells adhere to the fibroblast reticular cells in yellow, as you can see here in the animation, and migrate on the scaffold and are activated. They express a T cell receptor, ICAM1. The ultimate frontier in cell modeling is to model effects of a microvascular circulation. These are two possible approaches. One, here on the left, is to engineer the microvessels in vitro and anastomose them to the circuit that perfuses the culture medium. This has been done with encouraging results by several groups, but the regenerated vessels are still not stable in time, in terms of hierarchy and stability, to allow for physiological flow rates in the vessel's lumen. My approach instead is to implant the cell model subcute in a living organism, for example, the mouse on the right here. The main advantage of this approach is that the cell construct will be revascularized within a minimally invasive device, optically accessible in multiphoton. So the cell model can be implanted in a living organism, revascularized, and interact with the host immune system. We recently invented this concept of the microatlas, a miniaturized imaging window that can be used for intravital quantification of the host response to implantation of a biomaterial, a tissue, or administration of a drug. It's a miniaturized subcutaneous window for time-lapse analysis incorporating a scaffold for in vivo tissue regeneration. We obtained the first validation in vivo of our platform by implanting the microatlas in the corallantoic membrane of an embryonated chicken embryo cultured ex ovo. In the microscaffolds, we were able to visualize cells, the regenerated microvascular networks, and collagen with label-free 2-photon microscopy. Cell repopulation in the microscaffold is very fast and function of the scaffold pore dimension. So the microscaffolds are fully repopulated with cell density similar to the one present in unimplanted controls in only 4 days in the embryo. Most importantly, the geometry of the pore can be used to modulate the resulting cell density. Also, the same happens for blood vessels. We visualized effectively the microvascular network regenerated in the scaffolds in green autofluorescence by label-free two-photon emission, we found a significantly greater density of blood vessels in the scaffolds compared to the unimplanted areas. So the microscaffold can be used also to modulate the vascularity of the tissue regenerated by the host. And we saw that the microscaffold can be used also to modulate the properties of the fibrotic tissue regenerated by the host. So finally, we have implanted the microatlas subcute in mice, and this is what the system looks in two-photon microscopy with a developed moderate fibrotic reaction, and we can visualize in a label-free way the infiltrated cells in green, collagen, and the regenerated microvascular network in the scaffold. This is a two-photon zeta stack. So we're very excited about these results that show the potential of the microatlas as a valuable tool to face major problems of reducing, refining, and replacing animal testing in many fields, especially considering the low invasivity of this miniaturized window chamber compared to traditionally, and I would say ethically questionable devices. So in the end, I would like to acknowledge all the funding bodies that support my research, and I thank you for your very kind attention.